Today I'm going to teach, hey everyone, my name is Hope, and today I'm going to teach about the complement systems. So what you could be asked what a complement system means, it basically means a defensive system consisting of over 30 proteins produced by the liver. What I highlighted, those are like very important points you need to note down as a medical student. So they are found in circulating serum. So a complement system kills microbes in three ways. Microbes could be a bacteria, viruses, and all those sorts of kinds of things. So where I'm reading from is where I will highlight. Complement system kills microbes in three ways. There is opsonization. Opsonization is basically the process in the immune system where a pathogen, such as bacteria, are marked for destruction and phagocytosis by immune cells. Here I said 3B because as you'll see, in, for example, the classical pathway, you'll see that C3b is called an opsonin. It's called an opsonin because it's the one that actually marks these pathogens for destruction. So there are three ways in which the complement system kills microbes. It's opsonization, inflammation, and cytolysis. And inflammation is initiated by anaphylatoxins. This is the word here. The major anaphylatoxins we have are C3A, C4A, and C5A. But I will explain how these come about as we go up. As we go, one way I can remember it is 345. 345, and they're all letter S. 345, C3A, C4A, C5A, because that can be an MCQ. Which of the following is an anaphylatoxin? If it's C3A, correct. C4A, C5A. Then cytolysis, we shall see how the membrane attack complex is formed because it creates pores in the membrane of these pathogens, and then which finally leads to cytolysis. As you've seen here, I've mentioned it down, is that C5b, C6, C7, C8, C9 form the MAC. The MAC in full is the membrane attack complex. I've highlighted it there. So components are numbered in order of their discovery. That's how they were numbered. So we have components. The complement proteins are labeled from C1 to C9. I've written it here. C1 to C9. So remember that they were numbered according to the way they were discovered. If C1 was the first to be discovered, that's why it's called C1. C9 was the ninth one to be discovered, it's C9. So now the point where I say these components circulate in inactive precursor form, it means that they need to be activated for the cascade to happen. A cascade is basically activities that follow after another. Let me say, for example, let me give an example of how we as girls, when we're going to a toilet, it's like a cascade because I'm going to ask my friend, do you want to go to the toilet? Then she asks the other friend of the group. So basically, these complement proteins act like that. This activation of this one protein is going to lead to the activation of the other, to the activation of the other, until whatever you want is achieved, you know? And remember, we have seen there are three things involved. There is opsonization, inflammation, and cytolysis. So as we have seen, they're labeled from C1 to C9. I was reading from complement proteins designated by C, followed by numbers and letters. Some of you are going to be like, where are the letters from? For example, I've talked about the anaphylatoxins. Some of you will be like, where are the letters from? How did they come about? Yeah, I don't want this to scare you. So let's go to the next page. Where are the letters from? Remember, I've said we have complement proteins C1, C2, C3, C4, C5, C6, C7, C8, C9. Now, I want you to know some two special proteins. There is C2 and then there is C1. I want you to tag these, keep them at the back of your mind. So the first thing you need to know, remember, these numbers are in order of their discovery. That's point number one. They're in order of their discovery. Then C stands for the complement protein anyway. So they have said the letters. So whenever these proteins are cleaved, because they undergo cleavage, we're going to look at how the convertases do this. I'm trying to make these videos as short as possible. So I'm first doing the introduction, and then we shall look at the different pathways differently. So we have, let's say, for example, C1. The way I tagged C1 is because it's a unique complement protein. It's made up of C1q, C1r, and C1s. This is what it's made up of. And this C1Q has 6 subunits. We shall look at C1 deeper when we're looking at the classical pathway. I don't want to go into it, but let me look at the ones which are common to us. Let me use an example of C4. When C4 is cleaved, it's cleaved into remember we said there are letters C4A and C4B. C4A is the smaller fragment, and that's how it is for like C5, yeah. Actually, let me just say this. These ones are the ones that participate in this whole pathway things. These ones form the membrane attack complex. These ones here. Membrane attack complex. So this is a smaller fragment. This is the bigger fragment, or larger. Prefer to say larger though. Larger fragment. So why did you, I think someone is asking why am I saying C2 is special? So you see how I've told you that A is the smaller fragment and B is the larger fragment. B stands for big. One way to remember that. Why C2 is unique is that when it comes to C2, instead of A being the smaller fragment, it is the bigger fragment. And then C2B is the smaller fragment. That's the uniqueness of C2B. So let's go back to the introduction, and then we see what else we need to know. Then for the letters, A stands for the smaller fragment. I've already told you that. Larger fragment is B. These fragments are generated after cleavage, being broken into smaller parts. B is big. Remember, B is for larger fragments. There are exceptions you need to note. The exceptions you need to note, there is C1 and C2. As I told you, C1 is made up of, sorry about that. C1 is made up of C1Q, C1R, and C1S. C1S, QRS. C1Q is made up of 6 subunits, making it it's supposed to be making it able to bind up to 6 antibodies. We shall look at that in the classical pathway. What I've written here is what we've already gone through. So, general properties of the complement system, primary role. Sorry about the English problems. Primary role is lysis, which is a function of the membrane attack system. Remember, the membrane attack system is made up of C5b, C6, C7, C8, and C9. That's what makes up. Then the activity of the complement system is destroyed by heating up Sarah up to 56 degrees Celsius. But okay, this is something you're supposed to remember, but like I'm mainly here to do an introduction. Yeah. There's a pathway that is antibody dependent, and that's a classical pathway, as we're going to see. So we shall look at that in the next video. Yeah, in the next video. For now, that's it. Thank you so much for joining my YouTube channel. It was amazing having you here.